Today we are going to learn about gram staining, which is a very basic technique done in the field of microbiology to identify bacteria. So an introduction regarding gram staining. This is a method used to differentiate two bacterial species or a two group of bacteria or a two large group of bacteria called gram positive and gram negative bacteria, and this is the first step in identifying a bacterial culture or a bacterial specimen which is given to you. So when you get a bacterial culture in your hand and when you have to identify, the first step you will do is gram staining to identify whether it is a gram positive or a gram negative bacteria, and this is a very valuable diagnostic tool which is used in clinical and research fields also. So the invention of gram staining, and the inventor was Hans Christian Joachim Gram, and he is a Danish bacteriologist. He developed this technique to differentiate a gram positive and gram negative bacteria, and the technique was named after him as gram staining. Okay, so the aim of this procedure or this method or the staining method is to differentiate between a gram positive and a gram negative bacteria. So the principle, what is the basic principle behind this gram staining method is the cell wall composition of gram positive and gram negative cell wall. So the cell wall composition of gram negative and gram positive cell wall differentiates the two bacteria or the two groups of bacteria. Okay, so the cell wall of the two groups of bacteria, gram positive and gram negative, is the basic principle, or it is the thing with which you are differentiating gram positive and gram negative bacterial cell. So the difference between the gram positive and gram negative cell wall is the gram positive cell wall is having a very thick peptidoglycan layer, whereas gram negative cell wall is having a very thin peptidoglycan layer. Then in a gram positive bacterial cell wall, you have an additional components called teichoic acid, lipoteichoic acid, but in a gram negative bacteria you don't have those teichoic and lipoteichoic acid. And an additional difference between gram positive and gram negative cell wall is the gram negative cell wall is having an additional outer membrane which is absent in a gram positive bacteria. Okay, so gram negative bacterial cell wall is having an outer membrane which is completely absent in a gram positive cell wall. So how to remember? Usually we get confused, right? So we usually confuse the decomposition of a gram positive and a gram negative cell wall, or we confuse to differentiate both. So how to remember? You remember easily with the help of a mnemonic called long PPT. LONG, long PPT. Okay, so you think of a long boring PPT which you are compulsorily. So you think of a very long PPT which will be really a boring thing, right? So you remember the mnemonic long PPT. Okay, so long, what does it stands for? What the terms you have to remember for the long? So long, L stands for lipopolysaccharide. Then outer membrane, O stands for outer membrane, N for negative, G for gram. So you should remember like this: gram negative bacteria is having an outer membrane and also it is having lipopolysaccharide. So gram negative bacterial cell wall is having an outer membrane with the lipopolysaccharide. Then the next one, PPT. This you have to remember. P stands for positive. Again, P stands for peptidoglycan, which is thick, and teichoic acid. So PPT. Okay, so you can remember like this: positive, peptidoglycan, teichoic acid. T also stands for thick. Positive, gram positive, it is having a thick peptidoglycan and also it is having teichoic acid. So using this mnemonic, you can remember this difference between the cellular composition, the cell wall composition of a gram positive and gram negative cell wall. So the materials required for this, you need crystal violet, grams iodine, ethyl alcohol you need, and saffron. Then you need a microscope. Then you need a bacterial broth or a culture. Then you need glass slide, sterile glass slide. Then you need a inoculation loop to do this procedure. So how we will do this? What's the procedure for doing this experiment is, first you have to make a bacterial smear in the glass slide with the help of an inoculation loop. Okay, so the smear should be a transparent thing, or it should be transparent, so that the culture so that you can visualize the cultures easily. So the smear should be thin. Then after add drying the smear, you have to heat fix the smear. Then you have to add crystal violet, which is the primary stain. Okay, you are adding crystal violet, which is the primary stain. Then you have to add iodine, which is the mordant, or which is fixing the primary stain. Okay, so crystal violet you can add, then you are adding iodine, which is going to fix the primary stain well to the cells. Okay, then you are decolorizing the culture or the cell with the help of ethyl alcohol. Ethyl alcohol is the decolorizer which is used in gram staining method. So when you decolorize it, the gram positive bacteria is having a thick peptidoglycan, so it will not leave the stain, or it will. Since the gram negative bacterial cell wall is having a very thin peptidoglycan layer, it will leave out the primary stain, that is the crystal violet, and the color and the cells becomes colorless. Okay, so it is now free to take up the counter stain, that is saffron. Okay, so the gram negative bacteria is now taking up the color of the counter stain, that is saffron, and it appears pink in color. Okay, so when you observe it under the microscope, if your culture is a gram positive culture or a gram positive bacteria, the cells will appear purple in color or violet in color, whereas the gram negative bacteria will appear pink in color. So based on this difference between their cell wall composition, you can differentiate the two groups of bacteria, gram positive and gram negative, using this gram staining procedure. Okay. So again the basic steps: crystal violet will stain both the gram positive and gram negative bacteria. Gram iodine fixes the stain firmly to the gram positive bacteria, and ethanol or acetone you can use it for decolorization, and saffron is used as a counter stain, which is again it will restain the gram negative bacteria. Okay, so this is the basic step or the procedure behind gram staining method. So this is the diagrammatic representation. Okay, so you are staining with in a glass slide. First you are preparing the smear, then you are adding crystal violet, then you are adding iodine, then alcohol or ethyl alcohol, the decolorizer, then you are adding saffron. And so you remember the list of reagents you are using in gram staining: crystal violet, iodine, decolorizer, alcohol, ethyl alcohol, then you are adding saffron. Okay, so this is a diagrammatic representation of how do you perform a gram staining method. Okay, so this is how the microscopic view will be. Okay, so this is actually done in using a specimen. Okay, this is a medical procedure or medical specimen, medical sample. In this they have performed gram staining. So this is the image or a flow chart regarding the gram staining. Okay, and the result will be like this. Okay, so gram positive bacteria will be purple in color, gram negative bacteria will be pink in color. So this is a microscopic view of a gram positive and gram negative bacteria. So this will be the result. So when you have a purely, you are taking a gram negative bacterial culture, you can see pink color cells, and when you take a gram positive bacterial culture, you will see a pure purple or violet color cells you can see. Okay, so thank you. I hope you remember and you will understand the procedure, aim and the principle underlying gram staining technique. Thank you.